So, good morning, everybody. So sorry for the delay. We had some technical difficulties. Frau Schröder, I'm going to allow you to share your screen now.
Perfect.
Sorry that we have to do this over again.
No problem. Oh. I have. Yeah, here we go. Now you should see my presentation, hopefully. Or...
Yes, we do.
Perfect.
So wonderful. Again, I'm sorry about that. So we just said a few minutes ago, it was a bumpy road to this webinar. I'm very glad that everybody's here. And yeah, okay, I would like to start by, of course, welcoming you all to the webinar and to another episode of the PELOAcademy. Today we'll hear something from Dr. Insa Schröder about brain organoids, very fascinating topic. So we're very happy that this is happening. Let me introduce myself real quick. My name is Anna and I'm a marketing and management assistant at PELOBiotech. In case you have never heard of us, we are a cell culture specialist based in Martinsried in Bavaria. We are a small business with a huge portfolio of cells, media, and tools for cell culture. And if you're interested to check this out a little more, you can visit our website at PELOBiotech.com. You can find our catalog, our products there, but also a link to the PELOAcademy, which is a series of webinars that we've been doing in the past that you can watch on demand there. Very interesting things. Also, today's webinar will be posted there as well in a couple of days. If you want to go back, watch it again, or share it with anyone. All right, so yeah, for today, like even for me as a non-scientist, it is apparent how important 3D cell culture is becoming and organoids. We just talked about this topic, Frau Schröder and I. It's everywhere and it's very exciting, and we see that, of course, also in the turnout of registrations for this webinar. So let me present our speaker for today. As I said, it's Dr. Insa Schröder from the Helmholtz Institute. Now I have to read this. The Helmholtz Institute for Heavy Ion Research in Darmstadt. And Dr. Schröder is the group leader of the stem cell differentiation and cytogenetics group. So she started out in a different field from that. She's been doing food chemistry and toxicology, and very soon after her PhD that she took in these two things, she turned to stem cell research, and we might hear a little bit more about that. And yeah, so via different positions in the USA, as a junior group leader in Leipzig and the University of Halle-Wittenberg, she came in 2013 into her current position at the Helmholtz Center, yeah, where she's been ever since, and with lots of interesting collaborators, as I could read. Yeah, for example, the German Aerospace Center or the Federal Office of Radiation Protection, which to me was news that we have this office. All right, Dr. Schröder, please tell us more about your fascinating work. If anybody has any questions, feel free to use that question mark icon on the top of your screen, and I will read the questions to Dr. Schröder when she's finished her talk. So thanks again, everybody, for joining, and I'll pass over to you.
Thank you so much, Anne, for this kind introduction and also for the opportunity to share my enthusiasm for brain organoids. I'll take the opportunity gladly, and you will see that we cannot do without brain organoids. I have a disclaimer though. I can confirm that I have no personal or commercial link to PELOBiotech or any of their business, but I certainly have a link to these structures. This is one of the cerebral organoids that we produce, and you can see here the neuronal network in magenta with the nuclei here. So why are we interested in these organoids? Well, as you heard, I come from a Center for Heavy Ion Research. So heavy ions is an ionizing radiation form that is used for patients with brain cancer, and because it's a very efficient radiotherapy. But it is also a harmful thing in space because astronauts subjected to heavy ions will also suffer from alterations of their neuronal network and their brain. And what we, of course, can only observe, as the brain is so inaccessible, is, well, we have the patient's data like MRI, and we have epidemiological studies, we have animal models, and here we can see the detrimental effects on the behavior caused by radiation. But when it comes to the underlying mechanisms, mostly we relied in the past on animal models and 2D cultures. And I think I can convince you that the brain organoids are a nice add-on to find good countermeasures and implement new mitigants. And so the next minutes I will take to tell you what organoids are, how they are cultured and generated, how organoids help us to model brain development, neurological and neuropsychological diseases, and how organoids model brain cancers. And I will also briefly touch on a few challenges. This whole business of organoids is actually fairly old in terms of that there have always been cells that were used in 3D aggregates, but only when it came to, and I have to check now, I guess I'm stuck. Wait, here we go. Only when it was possible to isolate and culture human pluripotent stem cells, such as the human embryonic stem cells and the induced pluripotent stem cells, the whole business of organoids really went through the roof. And only from kind of 2013 upon, we have brain organoids. And so the first thing that you have to know is what is an organoid. And Wikipedia helps you very nicely, actually. An organoid is a miniaturized and simplified version of an organ produced in vitro in 3 dimensions that shows realistic microanatomy. So that's the two things, simplified version of an organ and realistic microanatomy. So an organ has to fulfill 3 requirements. It has to have multiple organ-specific cell types. It has to be capable to recapitulate at least some of the specific functions of an organ. And the cells are grouped together and spatially organized, similar to an organ in vivo. And you can generate these organoids from either pluripotent stem cells, and I will show you that, or organ progenitors, so primary cells. So what is not an organoid? Spheres are not an organoid because they are 3D, but mostly not organ-like because they are mostly contained of a homogeneous cell population. So a sphere of tumor cells, for instance, we use that to co-culture them or fuse them with brain organoids. But these spheres are not organoids. Nonetheless, they are equally as suitable. Organ on a chip is not an organoid, because you will put several different cell types together, and they may or may not assemble themselves as an organ, but it is not, you don't mimic the in vivo-like organ development necessarily. Nonetheless, this as well is very suitable. And I learned that even there are 2D organoids. Now, that is strange in the first place, because it is not really a 3D aggregate, but at some point, and this is the first paper that I could find that uses the term 2D organoid, sometimes it's really important that you, well, go back from a very complex organoid, dissociate them, and put them in 2D to see apical and basal polarization, for instance. And in this case, for a lot of virologists, this is of interest when they have to look at where does the virus infect the cell and how does its infection spread. So what's the origin of organoids? You will have a lot of organoids from primary cells, for instance. And here you see a whole selection. This is basically, you have the tissue, you dissociate it, sorry. You take the stem cells, put them in 3D culture, and get wonderful primary organoids, such as intestinal organoids or the likes. And they are used already for clinical streamlining of medications, for instance. But most of the organoids, and especially the brain organoids, start from actually pluripotent stem cells, meaning you have either human embryonic stem cell lines derived from a human blastocyst or human-induced pluripotent stem cells that you can get from patients via reprogramming. All these steps go through an embryoid body stage where you have all 3 germ layers, and then via a differentiation impulse, you get into the ectodermal lineage and into brain organoids. It requires some time, but you can also use brain organoids for a long time, for almost up to 2 years. But to get to a wonderful, good brain organoid, the starting material has to be very good. And this is what I want to show you here. You have to put a little bit of effort on your starting embryonic or iPS cells, because they should not only have good pluripotency traits and features and be able to be grown indefinitely, but they also have to have genomic stability, and they have to have the characteristics of being able to differentiate into all 3 germ layers. And especially for iPS cells, you should really make sure that what you think is in your dish is really in your dish, and need to do some identity checks. I want to show you one example where it really is mandatory, and that is genomic stability. So you have to have a karyotype analysis, and you have to do this karyotype analysis from time to time within the passages, because you can acquire genomic instability via culturing. And we do FISH analysis, so fluorescent chromosome painting, or you can also do the SNP analysis. Why is this important? Well, here you find an example of an iPS line, and this has a truncated chromosome number 5. Actually, this part is truncated, and I think it's added on to that second chromosome, because that seems to be too big. Now, chromosome 5, if you want to do brain organoids and check for something like a something that is dopamine or serotonin based as a mechanism, you should not have a truncated chromosome 5 in all your cells, because that chromosome contains the dopamine receptor or the serotonin receptor. So definitely with an IPS line that carries stable chromosomal aberrations, you should not continue. But if you don't have those, of course, you can generate brain organoids, and you can recapitulate the in vivo brain development from the very first start of the neuroepithelial cell that will develop into ventricular radial glial cells, the first progenitors that will then differentiate into the deep layer neurons. You will have the radial glia cells that form the intermediate progenitor cells, and in the end you will end up with astrocytes and oligodendrocytes, and you will get an areal identity. And this you can of course use to either get a brain organoid that mimics more or less the entire brain, or that only mimics parts of them, like the forebrain, the midbrain, or the hindbrain. And the classicals that you might know is the Lancaster protocol, and that you can see here is one that has lots of proliferations on neuroepithelial proliferation zones, and then towards the outer layer you find the more mature cells. And here you have examples of forebrain organoids, hypothalamus organoids, and midbrain organoids. And you can even do something like put these region-specific organoids together to form assembloids, and then you will have interactions between the two brain areas. And here you can see already that there are two ways to get to these organoids. One is an unguided differentiation. That's the Lancaster protocol that basically gives you everything. And then you have guided differentiation. And we in our lab actually use the two of them. We use the Lancaster protocol that starts off with the human iPS cells and ES cells. We use microfilaments to get a nice neural tube-like patterning. And then we only give a very short differentiation impulse and matrigel, and then we let them grow for up to 200 days and more. Here sometimes the limiting factor is the matrigel, and here I have to say, well, you all will know one company that is the main supplier, but here PELOBiotech actually also has an alternative for you that works equally as well. When you do this protocol, you end up with several of these, we call them proliferation zones, so the neuroepithelial cells that in the inside express the progenitor marker PAX6, and in the outside expresses the marker for neuronal differentiation MAP2. And if you go to a guided way, you can see down here with all the factors that you have to add in a timely manner, that these protocols are way more difficult or complex. However, what you get here is maybe a certain lineage preferentiation, or like in this case, we get not only the neuronal differentiation, but we also get nice glial cells like the GFAP positive astrocytes and also oligodendrocytes. And if you have these models, you can nicely model brain development with these cells. For example, you can mix wild-type pluripotent stem cells and pluripotent stem cells that have a DOX-inducible sonic hedgehog expression, and you can then mimic the sonic hedgehog gradient that actually leads to the brain development and the parts of the dorsal and the ventral brain. And you can do this here nicely, and you get these polarized organoids. This is a now my presentation is stuck. No, here we are. This is a work done by that has been done by Cederquist et al. And you can see they used a fairly low number of additions of differentiation factors, but the sonic hedgehog gradient already is nicely shown here. So sonic hedgehog is under the tomato reporter, so it's all the sonic hedgehog expressing cells are in red, and when you don't have doxycycline, you won't have the expression of the protein, but if you add doxycycline, then you get this sonic hedgehog gradient here, and that leads really to organoids that you can see here are nicely partitioned between the dorsal and the ventral part of the brain. And you can here nicely see regionalization of the brain. But organoids mostly interest us for neurological diseases, and one that I want to start with is modeling Zika virus-induced microcephaly using brain organoids, because it shows you how fast organoids can be used to figure out a disease. The Zika virus was known for a while, but there was a huge outbreak in 2015 in Brazil. And already in 2015, 2016, it was linked to microcephaly. So pregnant women gave birth to children with reduced brain mass. It was declared a public health emergency of international concern in February 16, and we had in March 16 the first human stem cell Zika virus model. And that was this because they could see that in brain organoids, when they infected these brain organoids with the Zika virus, what happened was that the SOX2 positive progenitor cells and stem cells of the brain were missing, so that the Zika virus definitely was interfering with the development. And we could see that as well in this study by Kayan, that what you have here normally, the progenitor cells, the neuroepithelial progenitors, and then the differentiation did not work in the Zika virus. And so the mechanism of this microcephaly could very quickly be shown in the organoids. Another thing that is also always a point for using brain organoids is something like the modeling tuberous sclerosis. Tuberous sclerosis is a rare autosomal dominant disorder that is caused by a mutation of either the TSC1 or TSC2. It leads to epileptic seizures, autism spectrum disorders, and intellectual disability. And What is known is that this disease or these mutations lead to cortical tubers, so cortical knots here, like shown like here, or also in this picture here. And it also leads to astrocytomas, so tumors, benign tumors in the brain, but that nevertheless also destroy the adjacent neuronal structures. But the pathological brain lesions that are seen in the humans, they are totally absent in the heterogeneous mice. So what is the heterogeneous humans show something that cannot be modeled in an animal model. And that is here a study by the Knoblich lab, and they could show that they can really model with brain organoids what happens in vivo in this disease, and that they can also, of course, then now look for countermeasures. But you cannot only model, let's say, early developmental diseases, but also things that you know all, like Alzheimer's disease. Again, here also, iPS cells from patients with familiar Alzheimer's disease or sporadic Alzheimer's disease were compared to patients with Down syndrome. And from these iPS cells, they prepared brain organoids, and they could see that you can generate in these organoids, what is generated is the amyloid beta plaques that you see in Alzheimer's disease. And you can also see in these patients the phosphorylated tau. So you have all the traits of the disease in this model, and again can use this for more studies, simply to find countermeasures or early biomarkers. And that leads me also to the topic that interests us the most, that is organoid models of brain cancers, or also of therapies that are used for brain cancers, I would say, because the first thing that we looked at was actually normal brain tissue reaction to radiotherapy. This is normally known in the field as brain necrosis or radiation necrosis. In the MRI, you see contrast enhanced lesions of the brain that get really big and lobulated over the time. If it's not treated, it can be lethal, and really nobody knows which patient will develop these lesions and side effects after radiation. And the really caveat is that these lesions occur sometimes very, very late after radiation therapy. So it's a late onset complication. The underlying mechanism is not known. It is supposed that the blood-brain barrier is involved, and that it is an aberrant angiogenesis, but it's not really clear what is the initiating point. So we set out to do our normal organoid protocol using the Lancaster protocol, and irradiated cells fairly early when they are cells are still developing, and late at day 80 when the cells were more mature. And what we found is something that looked like weird cavities. So what we usually have is a layer of double-edged lobes. Basically, that's normal. That are neuroepithelial proliferation zones that we want to see. What we didn't want to see and didn't know was these blobs, these blabbings, and they got really, really big and more in number with increased irradiation. And when we looked at the cryo slides and looked at markers for epithelial cells, we found that we saw what we see in the MRI, we also see in the organoids, forms that look like these lobes filled with fluid. And it really turned out that it was choroid plexus. These structures here are choroid plexus cells that produce cerebrospinal fluid. And this fluid is what fills then these cavities. And we could show that actually what might be identified as radiation necrosis, in fact, and we looked at necrosis, we couldn't find increased necrosis in those irradiated samples. So what we found was an aberrant choroid plexus formation that may lead to aberrant formation of cerebrospinal fluid that then destroys the brain tissue. And interestingly, what is done today as a treatment for these radiation necrosis is something that is actually termed to work against aberrant angiogenesis, so something completely different. However, the choroid plexus cells have the same receptors, so they will also, by accident, respond to this treatment. So this is something where we found a potential totally different way of identifying something as the cause of a phenomenon that has not been really well evaluated yet. And then, of course, we were also interested in, can we mimic a brain tumor? And here we went to the glioblastomas, because whatever you do, when you do surgery, chemotherapy, and radiation, the outcome is still that the overall survival is, well, maybe 2 years versus 1.5 years. So with all our sophisticated treatment options, these patients still don't really benefit. So what we did was a hallmark of glioblastomas is the overexpression of c-MYC, an oncogene. So we again went through our differentiation protocol in the beginning, but what we did was we nucleoaffected some of the early EBs. We transfected them with c-MYC under the promoter of and GFP under the promoter of using Sleeping Beauty transposase, and then we ended up with these tumor organoids, so random induction of c-MYC overexpressing cells, and we compared them to normal sister organoids, and we found out that this really mimicked a tumor, because these tumor organoids basically grew so fast that they overgrew all the organoids and destroyed them in a heartbeat. So we went to another model to take these organoids, sort the cells, and then transplant the GFP CMYC overexpressing tumor-like cells, either with organoid slices cultured in air liquid interface culture of the normal sister organoids, or fusing these tumor-like cells to little tumor spheres and fuse them to a normal sister organoid. And what we could see here was very interesting and something that we also did not expect really. The tumor spheres alone, when irradiated, were actually very sensitive and died very quickly, which is something that we don't really see in, or that we did not expect, because from in vivo study it appears that the stem cell-like cells, and these tumor spheres contain a lot of stem cell-like cells, that they are actually more resistant to radiation. However, when we fuse the tumor spheres to the normal organoids, they survived much better even after the 3 gray X-rays that we took here. And this is largely due to the organoid kind of interacting with the tumor sphere, and it's the astrocytes that actually grow into the tumor, and these organoids seem to protect the tumor spheres from radiation. And this is exactly where organoids are beautiful for, because you can monitor the interaction between the tumor and the adjacent normal tissue in a very nice manner. So this is something where I say organoids are a beautiful model, but of course organoids also have some challenges, and the challenges is that everything gets more complicated, more expensive, and it takes much longer. So of course you have a lot of different cell types that you ideally have to analyze in space and time, meaning you have a lot of immunofluorescent stainings to do, and this is of course more complicated the more complicated the actual organoid gets, and that means that there's a huge requirement for more microscopic procedures. So confocal microscopy sometimes will not help you anymore if you want to go into the depth of the organoid, but you may have to end up with light sheet microscopy, which leads to a lot of, well, challenges, because everything that you know from 2D will not really work in 3D and in the organoids. You may have to clear the organoids. Everything really takes way longer in these organoids, and almost none of the studies nowadays will be published without any RNA sequencing data, proteome data, etc., because only this will give you an overview of the various cell types that you have in your organoid and the various genes and pathways that are upregulated or altered in organoids after treatments and etc. However, having said this, this is still worthwhile the effort, because there's so much information that we can get from organoids that I think the brain organoids are still a very versatile 3D tool to study human neurological development and disease. And it's a good complement to, or a good substitute of, animal studies. And it's really a nice alternative to an otherwise inaccessible human organoid. And with this, I'm at the end of my presentation, and I'm happy to take questions and discuss the studies.
So thank you, Dr. Schröder. Can you hear me?
Yes, I can hear you.
Perfect.
Wonderful,
Okay.
sorry.
So thank you so much for this. We will move on to the question section, but I will take a moment to start a little Umfrage that we have planned, asking the attendees to answer this if they want to. And then, yeah, while this is running, maybe we can start with the first question that I have here. You talked a little bit about that already, saying things get more difficult and more expensive, but one question here is if you can get a little bit, if you can give a little bit of insight to the cost comparison to mouse models, for example. Yeah, whether obtaining data from organoids is very much more expensive, or is it really cost effective?
It is cost effective and maybe still more simple than animal models because apart from if you use embryonic stem cells, and in Germany you need to be allowed to do this, we have calculated that one organoid with all the costs, personnel and materials, etc., will cost us 15 euros. But you have the possibility to have 96 organoids in one go. And this makes it very attractive. And to be honest, I mean, all the funding agencies that come across my grant applications always look at the budget for the supplies and they're like, oh, well. If I then clarify it, they say, okay, yeah, that's okay. And they have not cut a lot of this so far. So yes, it is worthwhile.
All right, thank you very much for this. So another question would be, you mentioned microfilaments in the generation of the organoids. What are these microfilaments made of?
Yeah, this is actually suture material. So something that you find in any clinic. It's degradable suture material, and we cut it into little pieces and then dissociate it because it is a thread that is made of 6 single smaller threads. The idea about this microfilaments, and this was published by Lancaster et al., is that you get more oval shaped first neuroepithelial proliferation zones, and that helps the organoid to develop and to mature.
All right, thank you. So the next question from a participant would be, how can you keep genomic instability low while in cell culture? How does the use of different ECMs influence this?
Good question, very good question. You can keep it low if you really pass it strictly, and this is actually a learning process. If the if you have cultures that are a bit differentiated but still show nice colonies, then one is always tempted to isolate these colonies and then passage them on. But this is actually something where in culture you get very quickly very often genomic alterations. So I always tell everybody passage those iPS cells and ES cells regularly, leave them rather less confluent. So the smaller the cultures, the better. They will survive this. Pluripotent stem cells, good pluripotent stem cells will survive this, but the others will not. And so, but you cannot really, I mean, you cannot really avoid it totally. So you should always, well, once in a while, do a chromosomal stability test. And then if it does not work out, go to older batches. That's all you can do, unfortunately.
Thank you, Dr. Schröder. The next question that I have is, what are the applications of assembloids over organoids?
Assembloids are very interesting when it comes to diseases that require the interaction of, for instance, two different brain regions. So you can really, for example, depression is a disease where you see that the communication between the hippocampus and the forebrain is not as good as it should be. And in organoids, you can actually mimic this interaction. And so assembloids is always something that is interesting when you don't look within a brain region.
Thank you so much. We have so many questions coming in. That's amazing. All right. The next one is, are brain organoids also suitable for pain research?
I haven't. Well, I mean, you can of course enhance the generation of sensory neurons, but I would say at the moment I don't know of any study that is using it already. But well, pain, you would then it would be a functional test. You would only be able to say with the brain organoid, do sensory neurons function or not with an electrical input, and see whether they function or not. But to be honest, there I would see the mouse models or the animal models, yeah, to be favorable right now.
Thank you. And the next question ties into that maybe a little bit. So, can organoids be used to study cognition or diseases without physiological or macroscopical manifestation?
You can indirectly analyze cognition in that you can cognition, of course, is correlated with the interaction of the neurons with the number of synapses, etc. So that, of course, you can measure. And there are, of course, a lot of mutations related to cognitive decline or defects. And again, these you can study in the brain organoid, and this can lead to then pinpoint to mechanisms and maybe defects that then can be tested in a fewer number of animals. I think this is the point. You can use the brain organoids to streamline the next level, which would be the animal models.
Perfect, thank you very much. So in the study with radiation treatment, is there an increased intracranial pressure observed for patients? Do astronauts have more probability to succumb to this due to high energy radiation at that altitude?
Well, in the astronauts, it's a bit, you have the two impacts. I glossed over it a little bit, but of course you also have the microgravity. And the interaction of ionizing radiation and microgravity is the problem. With good shielding, radiation is not so much an impact as the microgravity is. And yes, in the patients, of course, that we see with this if you have an excessive cerebrospinal fluid production, that of course leads to the cranial pressure and to basically a degradation of the surrounding neuronal network if you don't treat it.
Thanks again. With the complexity that you showed in the papers, the question of consciousness comes to mind. Do these organoids exhibit similar neural waves or patterns to infants?
Oui.
I guess
no,
it's so. Do the organoids exhibit neural waves or patterns that are similar to infants?
I don't think so, but I don't know actually. I don't know the answer. No, they are way more. The brain organoids are way more primitive than the insects, and you don't have the input of what you would need to the brain actually.
All right, thank you. And then last but not least, a very interesting question, I think. Are there established guidelines or ethical considerations for conducting research on brain organoids, especially regarding issues related to potential consciousness? Is there research oversight? And what about the moral status of organoids?
That is a very important thing. I mean, as I said, consciousness is something where you have to have an input and you have to learn, and this is not what the organoids so far. It cannot be obtained by the organoids. So it's not, this term mini brain is actually wrong, because it's not a mini brain that we produce. It is an approximation to a neuronal network and to a brain as close as we get. But so far, no, we do not produce brain organoids that would have a conscience or could have a conscience. So, but this is a question that people working in this field often get, and we of course discuss it. So far, I have not seen a white paper on this. The white papers that we see in stem cell research is more about gastroloids, about embryos. That is more an issue, but certainly, I mean, what everybody tells you with brain organoids is no, you don't have to worry about any ethical relation to would this be a brain that feels and that can sense pain or anything yet. So that is not the case. Nonetheless, we have ethical issues, of course, using the cells from patients, etc. So we are, of course, need the ethical consent of the patients to use either primary cells or iPS cells generated from primary tissue or from the patients to be allowed to work with these cells and form brain organoids.
Yeah, that is indeed a complex issue.
Oh, yeah.
So one more question came in, namely, what do you think are notable highlights in the field and future directions?
Oh, good point. I mean, I think that any disease modeling, any understanding of disease mechanisms is still a big topic, definitely, and will be for quite a while, because I think that is something that helps us to also streamline the treatments and the drugs that are tested. So clinical research will be made more easy using organoids in general. And so that's one point. I have seen lots of, and this is why I have seen a lot of organoids, intestinal organoids, etc., made from primary patient tissue and used in preclinical studies. And I think that is still a huge field for anybody to go there. Yeah, I think that's it.
Wonderful. So yeah, a huge field for people to go into, you say. So maybe I can say something about the poll, where 50% answered with yes, they have been working with organoids before. 33% no, and 17% not yet, but planning to.
Okay, so then I hope I have not discouraged the ones that did not start yet, or that have not so much experience yet. And I hope that it was not too boring for the ones that are already working with the organoids.
No, thank you very much. I think that was actually a very exciting talk. Thank you so much, Dr. Schröder, for taking the time to talk to us about that. And yeah, for letting us have a peek into your work. So yeah, everybody, that concludes this webinar. Thank you everybody for participating and listening in. Have a wonderful rest of the day, and I hope we see each other soon.
Thank you.
Thank you, Dr. Schröder. Bye-bye.
Bye-bye.
So, hallo.
Hallo?
Schauen wir eben, wie es aussieht. Ein paar Teilnehmer sind noch drin. Gut, ich beende jetzt das Webinar und melde mich noch mal kurz telefonisch bei Ihnen, ja?
Perfekt. Vielen
Gut,
Dank.
vielen Dank.
Auf Wiedersehen.
Auf Wiedersehen.